Hello, my name is Raymond, and my poster presentation is on the persistent activity in the prefrontal cortex of the common marmoset during a delayed response task. Working memory refers to a cognitive system involved in the maintenance and manipulation of information over a short period of time, and it can be up to about 30 seconds. Subjects with schizophrenia are impaired on working memory tasks and fail to show normal activation of the dorsolateral prefrontal cortex. Cognitive abnormalities present in subjects with schizophrenia are similar to those with frontal lobe lesions. It has been found that reduction in the size of deep layer 3 pyramidal neurons in the prefrontal cortex of subjects with schizophrenia, and thus horizontal connections among pyramidal neurons in layer 3 of the dorsolateral prefrontal cortex may provide a critical anatomical substrate for working memory. One way of testing working memory is using a delayed response task. For example, during the cue period, food is placed in a randomly selected well visible to the animal. During the delay period, a screen is lowered and the wells are covered. During the response period, the screen is raised and the animal has to uncover the well containing the food. The figure on the right shows a schema of the firing patterns that foods are encountered in the prefrontal cortex that respond to different parts of the delayed response task, and that some prefrontal cortex neurons exhibit sustained activity during the delay period. This is regarded as a neural correlate of working memory. The common marmoset has attracted attention in neuroscience research because of its small size, high reproductive efficiency, and compatibility with gene editing techniques. This species' relatively less encephalic cortex offers practical advantages for laminar recordings, 2-photon, and calcium imaging. Marmosets has been established as a model for visual neuroscience, but its suitability for investigating higher order cognitive functions, such as working memory and its underlying neural circuitry, remains to be determined. It is unknown whether PFC neurons exhibit delayed period activity in the marmoset, as working memory in marmosets has been examined primarily at the behavioral level. We address this by implanting 96-channel microelectrode arrays into the prefrontal cortex of marmosets, and we use a spike gadget data acquisition system for untethered extracellular electrophysiological recordings during a touchscreen task. We used a delayed match to location task. A sample is shown in one of 4 locations. This appears during the delay period, and then in the response period, the animal has to select the correct location. Figure 1 shows the task accuracy and reaction times for varying delay durations. As you can see, the accuracy is higher than chance, and the reaction times are faster for correct trials than incorrect trials. Figure 2 is an exemplar neuron with sustained discharge activity during the delay period of the delay match to location task. The top 3 plots are raster plots. The y-axis is trial number and the x-axis is time. Each dot represents an action potential. In green is the raster for correct trials, and in red is the raster for incorrect trials. As you can see from the spike density function, there's an increase in the neuron's firing rate during the delay period of the task. Figure 3a shows where we implanted the array in marmoset 1, which is in area 46D, 46V, 8AD, and 8AV. Panel B shows the location of all identified units, and panel C shows the location of units with delay period activity. Here's a table showing the count and percentage of overall units modulated during different epochs of the task. About 39% of units are modulated during the delay period. Behaviorally, as expected, task accuracy decreased as a function of increasing delay durations. The neurophysiological basis of working memory has been investigated in detail in the prefrontal cortex of the macaque monkey. It is unknown whether prefrontal cortex neurons exhibit delay period activity in the marmoset, and thus our data show that prefrontal cortex neurons in marmosets exhibit persistent activity during the delay period of a delay response task, and that marmosets are a suitable model for investigating the neurophysiological circuitry underlying working memory. With a largely lysocephalic cortex and homology similar to macaques and humans, the marmoset is a promising alternative primate model for investigating the laminar microcircuitry underlying working memory in the prefrontal cortex. Ketamine treatment results in performance deficits in macaque monkeys and is similar to those seen in schizophrenia patients. The ketamine model can be paired with laminar recordings, and it has the potential to critically advance our knowledge of the functional connectivity of prefrontal neurons during cognitive tasks in healthy subjects, as well as patients with schizophrenia. Thank you for listening to my poster presentation.